Hello, hello. This is Nico, and this is a video where we're going to take a look, year 10, at how our DNA is actually involved in a process to make copies of itself. This is called DNA replication, and it is a very interesting process that goes some way to show you just how mechanical our DNA and the way our cells work. And it's kind of something over the coming lessons, we're going to be focusing on the different functions and processes that DNA is involved in before we get into how traits are inherited down from parent to child and so forth, down generations. So this is just a kind of nice little model kit showing the DNA replication process without showing any of the enzymes. Now, I wouldn't expect you to necessarily know what an enzyme is. It's a type of protein, okay? And this protein is kind of like the referee of chemical reactions in our body. It controls all of our metabolic function, a bunch of enzymes control all of our metabolic function in our cells. More specifically, enzymes tend to be highly specific to a single chemical reaction that occurs somewhere in our body. All right, and I've got a pictured model just trying to show you how they actually work. Most enzymes are typically a globular spherical shape. They work a little bit like a lock, and with the materials that they work on being similar to a key. All right. And what actually happens is we call the materials that the enzymes act on substrates, is that these small molecules that the enzymes work with, they dock into the active site, basically a little impression in the surface of the enzyme, kind of like a keyhole in a lock. All right. And that active site is usually very close to the shape of the substrate. All right, so they very much fit like a lock into a key. Now, one of the things we've learned in recent times is that enzymes, when the substrate docks into them, the enzyme tends to wiggle around a little and change shape so it better fits the substrate. And what this does is it changes the chemical environment around the bonds of the substrate and actually enables a chemical reaction to take place that is highly specific and often very unlikely to happen by itself in the human body. Okay, in this way, enzymes are often spoken of as being similar to catalysts, all right, in that they enable and speed up chemical reactions. All right, it is that enabling of chemical reactions that makes them more similar to catalysts. And again, that is something we'll explore in greater detail when we cover chemistry later in the year. All right, and from that, the product moves out of the enzyme, okay, and basically undocks from it and leaves. Now, enzymes can either break down materials, okay, like the one pictured is doing. It's breaking our substrate into two smaller pieces, all right, or they can build up materials, okay, and they can actually add small chemical groups onto an existing substrate and build a larger molecule. All right, they have a lot of functions, and we're going to explore some of the enzymes that are actually associated with the replication of DNA. So a big overview of what actually happens when we make a copy of DNA, when DNA replication occurs, is that we have, starting from the left-hand side of the above diagram, we have our strand of DNA, or more specifically, our two strands of DNA. Those strands separate, and then we build the complementary chain, the complementary strand alongside the old strand. And so we end up from two strands of DNA to two exact copies that conserve one of the original strands of DNA in them and a new complementary strand. that's not quite the mirror image, not exactly, because they are different nucleotides, but they do meet that A to T and C to G pairing of nucleotides. Now, this is all controlled by a big army of enzymes. There are many players involved. All right, we have helicase, an enzyme that unzips the chains. All right, we have a bunch of proteins that stabilize the chains called Ssb proteins that actually keep them separated. We have primase, which starts off the chains, so polymerase can come along and construct new DNA chains from free nucleotides in the cell. And polymerase is actually a whole group of enzymes. Polymerase 3 is the one that actually builds the chains. Polymerase 1 and 2 do a variety of functions, including the proofreading of the new chains being constructed by polymerase 3. Okay, and they actually fix any errors we have proofreading and error correction in our DNA. And then we have another enzyme called ligase, which comes along and rejoins the strands, fragments of the strands from small pieces up to the larger overall strand. Okay, and you'll see in a second why that's important. Okay, so just kind of like just a representation of it. All right, we have helicase coming in, and I'm just going to turn annotation on. All right, so we have our helicase coming in and opening the chain up. All right, we have our primase attaching RNA to the open strands of the existing DNA. We have our stabilizing Ssb proteins. You can see them beside the helicase. And then we have two different things occur. On one of the strands, polymerase 3 attaches and just builds along. Okay. And on the other strand, what happens is polymerase 3 attaches and builds the strand in sections. Now, the reason for this, if you recall, the structure of DNA, the backbone of the sugars and phosphates, has two different directions that it is actually running along, the 5 to 3 and the 3 to 5 direction. And these enzymes can actually only go in one direction. So the way our cells get around this is they actually have one strand go along in a straight line and the other strand basically loops out and joins them together. You'll see that in another resource I'll link you to that will show you a lovely computer animation of all of that. So just kind of in summary, all right, and a good place to start is to focus in on a relatively simple molecule, simple organism, the E. coli bacterium. All right, and that is we start with by having the helicase enzyme that's represented by the triangle in the parting chains. That opens up the DNA at the replication fork, and all this kind of looks like a bubble forming in the strand of DNA. All right, at that point, we have our single strand binding proteins, our Ssb proteins. They coat the DNA around the replication fork to hold it open and prevent the strand from rewinding together. All right, they've got to keep those hydrogen bonds apart. All right, they have a topoisomerase. I can never say that one correctly. All right, they have an enzyme that works just past the replication fork to actually avoid the DNA from forming some of the larger coiled structures that the strands form. All right, basically if you think of it as being like a plastic ruler that is twisted around and under tension, all right, that enzyme is actually lowering that tension so the DNA doesn't coil up and spring back into shape. And then if we take a look, I'll turn annotation back on. We have our primase, which I'm just trying to find one just here. Here we go. Our primase joining just here. All right, and putting in some RNA bases to actually start the polymerase off. And one of those strands will actually form in the 5 to 3 direction, and that'll just go along in a straight line. The other strand, which is the lagging strand that loops out from the replication point, okay, actually has the DNA constructed in patches along it. All right, these are called Okazaki fragments, named after the pair of scientists who discovered them. What happens is the DNA ligase runs along those chains and actually removes the RNA primers and joins them back up together. Okay. All right. And then once all of those nucleotides are fitted to the existing chains, the enzymes detach from the chains and the DNA coils back up together. All right, so all of that happens with clockwork-like precision. Okay, and one of the things I wanted to just highlight just here is that we've kind of got a really good understanding of how all of this works, to the point where, you know, we know how these nucleotides exist as free nucleotides in our cells. They actually have the nucleotide, so we've got our nucleotide just here, and we have our sugar phosphate backbone, which actually has a triphosphate group on it that gets, this bond here gets actually broken in the process of joining it to the new chain. And the leftover energy from breaking that chain actually powers the entire process. All right. And so all that kind of just tells you about how the whole process actually works. All right. And what actually the bonds that are formed are called a phosphodiester bond, which is eliminating a small molecule, the phosphate ions being released from it. And it's not dissimilar to the reactions that occur to actually assemble other molecules in our bodies, like fats and so forth. All right. Okay. And it's something we'll explore more in the chemistry topic when we look at ester bonds. Now, finally, I would like to just point out that the process that occurs in E. coli, so a prokaryote, all right, a slightly simpler organism than most multi-celled organism, it's a slightly simpler replication process. However, that is not to say that the E. coli is not a highly evolved organism. It's been evolving just as much as us. It's just fitting its niche as a single-celled organism. All right, eukaryotes, these are organisms that have membrane-bound organelles. All right, multi-celled organisms like us humans, like plants and so forth. All right, they tend to have multiple linear chromosomes. So they have multiple strands of DNA that are packaged up around proteins inside their nucleus. All right, so that means that to actually copy those massive amounts of DNA, all right, it is quite possible for multicelled organisms like humans to have 100,000 active points of replication, okay, that occur across their DNA when it is copied. All right, and the other thing, of course, to point out is most of the enzymes that actually are involved in the replication of DNA in E. coli have a counterpart for eukaryotes like us. All right, but sometimes a single enzyme that is represented in the model that we just discussed for E. coli might have multiple enzymes in a eukaryote like us. Okay, for example, we talked about 3 polymerases, and in humans there's at least 5 that have important roles in replication. All right. Now, because most of our DNA is linear, what ends up happening is that by having the lagging strand, some of the DNA tends to be lost from the end of the chromosome. This is called the telomeres, okay, when we replicate our DNA. Now, the shortening of these sections of our DNA has actually been linked to aging as a multicellular organism. All right, and so we can kind of think of this as being a molecular representation of the process that occurs in terms of replication of our DNA, and think of DNA replication as being similar to taking a photocopy of a photocopy of a photocopy of a photocopy. Part of the reason why we age and we begin to break down as we age is that our DNA is actually getting shorter. And that explains why we have a finite lifespan. All right, there is more to it, and I'm talking very briefly about it. All right, so it's perhaps something you can explore in your own time. And finally, I would like to stop on a 3D image of a model of this side of replication. Now, the colored blobs in this image are representing at least part of an enzyme, most likely just the active binding site where the enzyme is bound to the strands of DNA. The right-hand side is the leading strand, okay, continuing off in a straight line, and the upper section is the lagging strand, which is looping back round. Alrighty. Thank you for watching the video, Year 10. Let's continue with the lesson. Bye-bye.